I'm honored to be able to present on behalf of all the study co-authors the results of this ETCTN study led by Dana-Farber of the fibroblast growth factor receptor inhibitor rogoratinib in patients with succinate dehydrogenase deficient gastrointestinal stromal tumor, or SDH deficient GIST. GIST is the most common sarcoma of the GI tract, and the majority of cases of adult GIST are driven by mutations in the genes KIT or PDGFR alpha. However, for about 10 to 15% of adults with GIST, and for most patients, pediatric patients with GIST, the GIST is characterized rather by a functional loss of the SDH complex. This results in DNA hypermethylation, leading to an upregulation of signaling through the fibroblast growth factor receptor, or FGFR, which can be a known oncogenic pathway. Blocking signaling through the FGFR pathway with FGFR inhibitors has been shown to have clinical efficacy in other types of cancers, like cholangiocarcinoma or urothelial cancers. In preclinical experiments in SDH-deficient PDX models, SDH tumor growth was suppressed with the use of rogoratinib and pemigatinib, which are both FGFR inhibitors. This was a phase 2 study conducted at 11 U.S. centers of the FGFR inhibitor rogoratinib in adult patients with SDH-deficient GIST, a type of GIST for which effective therapies are greatly needed. In the study, 24 patients with unresectable or metastatic SDH-deficient GIST were treated with rogoratinib. 10 patients, or 41%, had a partial response, and another 12 patients showed disease stabilization. While SDH-deficient GIST can sometimes show an indolent growth pattern, prior studies of drugs in SDH-deficient GIST reported median progression-free survivals of only around 9 to 12 months. In our study, however, responses were durable, and 9 patients still on study at the time of the data cut. The side effects from treatment with rogoratinib were overall mild to moderate and were manageable with dose reduction if needed. Toxicities were typical of the FGFR inhibitor class, with hyperphosphatemia as the most common side effect. The results of the study are remarkable for a response rate that is significantly higher than has been reported in prior clinical trials in SDH-deficient GIST with other investigational agents. This study demonstrates the critical impact of epigenetic alterations in the pathogenesis of SDH-deficient GIST and the effect of targeting the resulting aberrantly activated FGFR signaling pathways.